Swayam Prabha. Digital India. Educated India.
Welcome back. So in the last class we had learnt about the theory for how to design a batch reactor system, and we will see how to find the solutions for those problems. So let's begin with the first problem. Can you see the problem on the slide? Now let's read the problem. Single cell protein is produced from methanol using a 1000 meter cube bioreactor. So here methanol, I hope you can understand, will be the substrate, and the volume of the bioreactor is also given to us, which is 1000 meter cube. The biomass yield, which is of the single cell protein from the substrate, is given to us as 0.42 grams per gram. The Monod saturation constant, Ks, is also given as 0.7 milligrams per liter, and the maximum specific growth rate is given as 0.45 hour inverse. The medium is said to contain 4% methanol. So which would mean what? As it is weight by volume, so this corresponds to 40 grams per liter of methanol, and a substrate conversion of 0.98 is desirable in the process. So what is desired is 98% of the substrate by the end of the batch process should get consumed. Now for batch mode of operation, the initial cell concentration is 0.1 grams per liter. So this is given to us, and the downtime between the two successive batches is said to be 5 hours. So generally downtime means the time which is spent in harvesting, then re-preparing, sterilizing the reactor for the next run, cleaning, all that comes under the downtime till the next reactor run is ready to start. So what has been asked is, first, calculate the annual biomass production. So let's see how can we calculate the annual biomass production first. So first we need to calculate total mass of the cells which will be produced during this batch culture. Now the total mass would be what? Then of course the mass of the cells which will be produced should be, during the batch process, will be the difference between the final and the initial biomass concentration multiplied by the volume of the batch. Now to find out the batch time for the culture and the total time which is needed before you run the next batch will be the sum of the batch time and the downtime. Then only you are ready to run the second batch. So the total time which is required, or which is the gap between the two batches, will be the batch time plus the downtime spent for the one batch effectively. So the number of batches which can be carried out in 1 year can be calculated as this, given in equation. 1 here, 365 the number of days, 24 the number of hours in each day, and total time given as T with a subscript capital T. So this will give us the total number of batches which can be run in an entire duration of 1 year now. So now the annual biomass concentration which then can be produced would be equal to, so annual means in 1 year. So in 1 year then how much amount of biomass will we be able to produce? The biomass produced in the 1 in one batch multiplied by the total number of batches which are possible in a year. So, this is what is given here. Now, we need to calculate the annual biomass production. So, how do we do that? What is given to us? The initial substrate concentration, which I said corresponds to 40 grams per liter of methanol. Now, the final substrate concentration is also known to us, because if you remember the problem, it was given that 98% of the substrate is to be converted desirably. So, if 98 gets consumed, so 2% remains. So, the residual substrate concentration or the final substrate concentration in the broth would be 0.8 grams per liter, as done here after 98% conversion of the substrate. Now, if the initial biomass concentration was given as 0.1 grams per liter, we can calculate the batch time. It is given as 11.67 hours. How? Now, your batch time can be calculated as given in this equation, which I have underlined on the slide. Any guesses how we have reached to this equation? So, let us see that. The culture is following Monod's kinetics. Now, if you remember the problem and if you see the problem carefully, the value of Ks is very, very less than the initial substrate concentration, which is given as in the orders of ppm, whereas the substrate concentration is in grams per liter, the tens of, which means 40 grams per liter. So, if the Ks in the Monod's model becomes very, very less than the S in this equation, if Ks becomes very, very less than S, then we sometimes can neglect Ks. So, then your mu tends to mu max. Now, we know Yx by S is equals to x minus x naught divided by S naught minus S, where x naught and S naught are initial values of substrate and biomass and substrate concentrations. So, now you can relate x with S. Let us see how do we relate. Yx by S multiplied by S naught minus S plus x naught should be giving as x. So, I think this is pretty simple. Now, x we know is an exponential function of time, isn't it? This is the exponential growth. So, your ln x by x naught is equals to mu into t. Now, this mu here is nearly equal to mu max. So, now your batch time can be given as 1 by mu max multiplied by ln x by x naught, isn't it? Now, here further the x can be substituted as the equation 1 here. Let us call this as equation 1 and this as equation 2. So, if we do the substitution of x in terms of the entire portion which I have put in curly brackets in equation 2, then you can calculate the value of batch time, because rest of everything in this equation is known. We know S, we know S naught, we know x naught. So, everything is solvable. So, this becomes your batch time. This is what is given. This is what was given here as well as this equation. So, if you expand this, you will reach where we were. This is x naught plus yx by s, s naught minus sf divided by sf is the final concentration, which we were calling as S, versus x naught. This is going to give you the batch time. So, if you now put up all the known values, then you will get an answer of 11.3 hours. Now, once you know the batch time, then you can calculate the final biomass produced by the equation given in the end, which is the exponential growth rate, how the biomass changes as a function of batch time. So, at the end of the batch time, you will substitute T in the form of batch time, and the culture is growing at its mu max. So, the initial biomass is known, so you can calculate the final biomass concentration. So, it comes out to be 16.5 grams per liter. Now, the next part of the question, it is asked how much total mass, actually it is the same part. So, we had to calculate the annual biomass produced using this process. So, the total mass of cells which can be produced from this batch culture will be, now you know the xf, you knew x naught, and you know the batch volume. So, now you can calculate the mass of cells produced after each batch. This turns out to be 16,400 kgs. Now, the total time before we run the second batch will be the batch time plus the downtime. Now, we know the batch time, and we have also given in the equation, we have been given in the problem the downtime. So, we will add the batch time with the downtime, and it turns out to be 16.3 hours as the total time. So, the number of batches which can be carried out in 1 year will be 365 into 24 divided by the total time, because your batch time is in hours. So, it turns out that the batch time, the number of batches which can be run annually will be 536. So, the annual biomass production can be now calculated. After one batch, we knew from equation 1 on this slide how much could be produced till we run the second batch. So, now if you know the total number of batches which can be run, and then you can multiply it with the amount of biomass which you will obtain after running one batch, you will get the total amount of annual biomass produced, which is 8,790.4 tons per year. In the second part of the question, it was asked what size of the reactor should be chosen if the demand of the biomass produced is raised to 10,000 tons per year. Now, to solve the second part, the reactor volume given for the batch process was 1000 meter cube. Now, if 1000 meter cube reactor is giving us from the first part 8,790.4 tons per year, so 8,790.4 tons. Then what volume will be needed for 10,000 tons. Both are per year basis. So, that is a straightforward problem, and then the answer is 11,037.6. Let us see another problem. A strain of E. coli has been genetically engineered to produce human proteins. Okay. A batch culture is started by inoculating 12 grams of cells into a 100-liter fermenter containing 10 grams per liter glucose. So, what all is given to us? They have given us the initial cells which have been inoculated, the starter cells, the volume of the fermenter. So, we can calculate the initial biomass concentration, isn't it? The amount is given to us and it is 100 liters. So, it turns out to be 0.12 grams per liter, containing 10 grams per liter of glucose. So, the substrate initial concentration is also given to us, which is 10 grams per liter. The maximum specific growth rate of the culture is given as 0.9 hour inverse. Okay. So, mu max is also given to us, which is 0.9 hour inverse. Now, it is also said to assume that the culture is growing at its maximum specific growth rate in the exponential phase. So, which means that your x is equals to x naught e to the power of mu t in the exponential phase. So, now here mu can be replaced as mu max. The biomass yield from the glucose is given as, which means Yx by S, which is given as 0.575 grams per gram of glucose consumed. Okay. They are asking us to calculate the time required to reach the stationary phase. So, students, if you remember, when does a culture reaches the stationary phase? If you remember the batch growth curve, it starts, the exponential phase starts and it reaches the stationary phase once the substrate is depleted, and this is our exponential phase, and the culture is growing at its mu max. So, we can assume it is a constant slope. So, this is our mu max, and so now we need to calculate the time required to reach the stationary phase. So, during the exponential phase, as I had written earlier, it is a function of exponential function of time. So, now xf will be equal to x naught, xf stands for the final biomass concentration, mu max multiplied by the time. What time to reach the end, the maximum biomass? This is what has been asked. Let us call it as the batch time, TB. So, now how do we calculate the time? Any guess? Right. So, if you remember, it will reach the stationary phase when the substrate is consumed completely. So, there is substrate depletion, then only it will reach the stationary phase. So, you can assume that the substrate, the final substrate concentration is nearly 0. Now, Yx by S is given to us. So, now Yx by S and what all is given to us? How it relates the x and s? The xf minus x naught divided by s naught minus s. S naught is given to us, s is nearly 0, x naught is given to us, xf we need to calculate, Yx by s is also given to us. So, now equation 1 can give us the value of the final biomass, isn't it? Now, from the equation 1, if we calculate the final biomass concentration, from equation 2, we can then calculate the batch time or the time required to reach the end of the log phase, and then we will be able to answer the part 1 or A. Now, let us see part B. What will be the final cell density if the fermentation is stopped after only 70% of the substrate consumed? Okay. So, generally for part A, what did we assume? It was said that till the end of the log phase, the reactor has been run, which means so that we could assume that the substrate has completely depleted. Now, they are saying rather than assuming that, what if the reactor is harvested when the final substrate concentration is 30% of the initial substrate concentration, then only 70% of the substrate is consumed. So, now you know the initial substrate concentration and you know the desired conversion, which is 70%. So, now you can calculate the final substrate concentration. So, now what is given to us? The initial substrate concentration is given, the final substrate concentration can be obtained after 70% of the desired conversion of the initial substrate. Yx by S is also given, and we know how Yx by S relates the biomass production with the substrate consumption, Xf minus X naught divided by S naught minus Sf, isn't it? F stands for final, O stands for the initial value. Now, if this is known, Sf is also known, X naught is also known, we can easily calculate the final biomass. Let us see the solution now for the first problem which we discussed. X naught we can calculate given the volume and the mass of cells which are added in the reactor. So, the concentration at time t is equals to 0, when the cells start dividing, it is or start getting into the exponential phase can be assumed as is 0.12 grams per liter. Now, we need to calculate the time when the culture reaches stationary phase. So, here we are saying that the final substrate concentration is 0 at the start of the stationary phase. The culture is growing at its maximum specific growth rate, and from the earlier problem, if you remember, when we used the two equations, x is equals to x naught e to the power of mu t and yx by s is equals to x minus x naught divided by s naught minus s. And in the initial problem, we used these two equations, and here mu was again can be assumed as mu max. Then we could relate the batch time as a function of the substrate concentrations and the initial biomass, because the final biomass is not known. So, there can be more than one ways to solve a problem. So, here you can calculate the batch time after putting up the known values in this equation. Now, to solve the second part, if only 70% of the substrate is consumed, the final substrate concentration would be 0.3 times S naught, 30% of the initial substrate concentration, which turns out to be 3 grams per liter. Now, if you substitute this SF value in the previous equation, here you can calculate the value of the time which will be required for 70% conversion of the substrate. So, once you know that time of batch for 70% conversion of the substrate, you can then substitute in this equation and find the value of final biomass concentration obtained, which is this value.